Hi everybody, it's Mrs. Mancuso doing a little bit of AP Bio review, and I want to talk about transformation. To be transformed means that you are different, and so transformation is something that bacteria do, and they take in DNA. And so to be transformed means to take in DNA. And this is pretty unique. Humans do not do this. A bacterial cell, so here's its cell, it has one chromosome. And the one chromosome tends to be circular, which means this is one strand, like how we have one strand, but it actually curves around and connects. So that would be its one chromosome. Doesn't have a nucleus because it doesn't have membrane-bound organelles. So that's just its one chromosome. It actually has extra little chromosomes, and the extra little chromosomes are called plasmids. And we have found that on plasmids are resistance genes. And we treat bacteria with antibiotics. So antibiotic resistance is a gene that exists in bacterial populations. So if you go and you're sick and they say, well, you have this bacterial infection and the doctor gives you a prescription for amoxicillin, and you take amoxicillin and you're not getting better, it might be because your bacteria are amoxicillin resistant. They have this gene. Well, the cool thing about bacteria is they can do this. They can share their plasmids in the environment, and another bacteria can pick it up. Now it's resistant. If it transcribes and translates that gene, it now has the same resistance. This passing is called transformation. So we did a lab where we transformed E. coli. So we had E. coli and we heat shocked it. And when you heat shock something, you make it more likely to take in the plasmid. You make the chances of transformation increase in percentage. So we treated our E. coli with a plasmid, so a little circular piece of DNA, and that circular piece of DNA had two genes on it. So I'm going to have one gene be here. This is my P-glow gene. That is for green fluorescent protein. So I gave it a piece of DNA that would allow it to glow as long as it had the activating sugar. So if you have the activating sugar, you can glow if you get this gene. And over here, we gave it another gene, and this is an ampicillin resistance gene. So here's our E. coli. It took in this plasmid, and if it transcribes and translates it, it will be able to glow and it will be resistant to ampicillin. So we actually had 4 plates. Really common for the AP to show you this and ask you questions about it. So I want to make sure you really understood what we did. These are 4 Petri dishes. The Petri dishes have agar in them, and the agar is basically food for the bacteria. We kept it at an incubator at 37 degrees because that's the same as body temperature. So we know it's a great temperature for bacteria to survive. This plate was just luria broth. That's it. We didn't give it, I'm going to put negative because we didn't give it the plasmid. We just put luria broth on there. And we actually saw this. Our bacteria grew like crazy. That's because why wouldn't it? We gave it food. We gave it warmth. It grew. It didn't matter if it had a gene. There was nothing that it needed a gene for. This is just growing bacteria. Then, so this is a control. You do a control to make sure that your bacteria will actually grow in the conditions that you're giving it. If there's something like, oh, maybe your incubator was not working and it was saying 37, but it was really 67 and they all died. Well, you'd know it because they wouldn't grow on this plate. So it's important to have controls for that reason. All right, I have another negative one, and that's luria broth and ampicillin. So if I have luria broth, that's food. If I have ampicillin, that's an antibiotic. If I did not give my bacteria the plasmid, no growth. Nothing should grow on here because the ampicillin will kill them. Now I could have luria broth and ampicillin and a positive. Positive means I gave it the gene. I should be able to see colonies growing here. Those colonies are showing transformed bacteria. So every colony, this is 100% transformed. Okay, not everyone took it in. So this is not every bacteria from here, but every bacteria colony that you see is transformed. So all of these took in the Ampicillin resistance gene. I don't know if they glow or not because I do not have the sugar present. So I can have another one that's positive, and it has Luribroth, and it has Ampicillin, and it has arabinose. Arabinose is the activating sugar that you need to turn on this green fluorescent protein. So again, I'm going to see colonies, but all of these colonies are going to glow. And that's because they are expressing the green fluorescent protein gene. So you should be able to look at what the conditions of a plate are, look at what was taken in, and determine what will be expressed. You only see colonies, not lawns, because transformation is not a 100% thing. Not every bacteria is going to become permeable to the plasmid and let it in. Only some will let it in. All right, thanks for joining me on Transformation Review.